I know that I remember that I did not finish this particular topic. So I hope it's fine with you, correct? Yeah. So, so let's go with our topic, that is chromosomal disorders. So in Mendelian disorders that we saw in the last class, so we understood that the genes which are defective follow the Mendelian Mendelian genetics. So those disorders are called as Mendelian disorders. Now, what is the difference between Mendelian disorders and chromosomal disorders? Is the first common question that comes into our mind. So if you have this question, it's very simple to answer this. So if you see chromosomal disorders, there is a bulk load of physical traits that you can observe in a particular individual. So for example, if you take, I know that you have learned this lesson. If you take Down syndrome or Klinefelter syndrome or Turner syndrome, you don't pinpoint to a particular, effect that you see in their body. So you have multiple effects. For example, if you see Down syndrome, for example, it is an autosomal trisomy disorder. So you can see that there is a lot of physical traits or phenotypic characteristics that you can look into this particular trait. If you see Mendelian genetics, you are looking at one character or one particular gene. So now what is happening? The gene behaves in such a way that it follows law of dominance, law of segregation, or law of independent assortment. But here it does not happen like that. Some people get it, some people don't get it. So the reason would be the chromosomes for the defects. So that is a major difference between Mendelian disorders and chromosomal disorders. So first we'll discuss about chromosomal disorders. In chromosomal disorders, so when do we call it as a chromosomal disorder? See guys, if you look at any particular situation, for example, you know, why a particular person will have a defect in their chromosome. So these situations where you don't have a healthy number of chromosomes or healthy shape and size of the chromosomes, this is called as chromosomal aberrations. Okay, I'll repeat the term. The term is called as chromosomal aberrations. So what happens in chromosomal aberrations? In chromosomal aberrations, an individual, okay, will have abnormal number, abnormal, haploidtic or diploidtic types, or one homozygous is extra, or one, in homozygous pairs, it will be only one monosomy or trisomy conditions you can see. Or when you see in the structure of the chromosome, it will be a little bit of defect in the size or in the number of genetic material that it has to contain. So these are called as chromosomal aberration. So the major cause is, you can pinpoint to many particular situations. One common defect that you can see is whenever a couple who are very very aged, for example 35 years above, or around 38 years above, let's take it like that. So if you see that when these couples marry, or whenever the elderly couple they try to make kids, during the gametogenesis, what happens? The spindle fibers that forms will not be as healthy as the younger ones. So the segregation of chromosomes, there is a defect in how it segregates. So there is a high risk in aged couples where you can see the chromosomal aberrations. So this is the most happening factors, because other factors are also there. For example, exposure to radiations, exposure to or genetic history, etc., etc. But for a healthy individuals, for them to get a chromosomal aberrations is due to because of the aging factor. So that's why that we have to keep it in mind so that we can understand this concept. So first we'll discuss Down syndrome. What is Down syndrome? A Down syndrome is, for example, if you see a normal human being, we know that we have 46 chromosomes in the diploid state, or we can call it as 23 pairs of chromosomes, correct? So this is how it is in all the healthy individuals. But in Down syndrome, what happens is we have 47 chromosomes. Okay. So how did they end up with 47 chromosomes? Is, see, 44 chromosomes are called as autosomes, and the last two chromosomes, whether it is XX or XY, these both are called as sex chromosomes. Correct? No, these are sex chromosomes. These are called as autosomes. Okay. So many of you know that what is an autosome and what is the role of an autosome. But let me tell you, for some people who don't know what is an autosome, what is a sex chromosome, so this will help you to understand. Guys, look here. When you look at a human body or any living organisms, you can pinpoint to two different categories. You can, when you look at them, you can pinpoint to two different categories. One is the vegetative characteristics, other one is the sexual characteristics. For example, if you look at a lion, you can differentiate between a lion and a lioness, as simple as that. So the ability for you to differentiate between lion and a lioness comes from the sex chromosome. Simple, right? Whereas the rest of the characteristics, that means both lion and the lioness has the eyes, the nose, the hair, the fur, the legs, the limbs, the characteristics of a lion, all of them are very similar between both of them. Only the sexual characteristics are different. The one that gets their lioness and lion character is from their sexuality, and the sexuality comes from the sex chromosomes. Whereas if you look at the other part, that is the vegetative characteristics, the number of eyes, the type of eyes, the nose, the behavior, the abilities, all of them are called as autosomic characteristics or called as vegetative characteristics. So these vegetative characteristics, even I, you, and every living organisms have. So we can divide the chromosomes into two. One is the autosomal set, other one is the sex chromosome set. If you see the autosomal chromosomes, the autosomal chromosomes lie between 22 pairs, correct, right? And the last pair, that is the 23rd pair. So the 33rd pair is called as a sex chromosome. Agree? So now what is happening, guys? These 22 pairs, any chromosomal aberrations that happen within these 22 chromosomes, they are called as autosomal disorders. So when you look at the Down syndrome, this is called as a karyotype. This chart is called as a karyotypic chart. So what is happening is this is karyotyping is a technique where you understand the morphological characteristics, that is the physical nature of how the chromosomal shape, structure, and number is. Based on that, we can determine the health. So if you look at the karyotype of an individual, what you can see is the 21st pair of chromosomes. Oh, sorry, guys. The 21st pair of chromosome is not normal in Down syndrome. So what is happening here in the 21st pair, instead of homologous pair, that is, if two chromosomes are located like these, okay, this should be 21, 21, right? There is another 21 chromosome. So this 21st extra chromosome is called as a trisomy condition. Okay. So what kind of trisomy we are experiencing? We are experiencing autosomal. Okay. It is an autosomal trisomy. So in autosomal trisomy, what do you have? You have an extra chromosome along with a homologous pair. You see here we have 21, 21, 21. Other pairs are only 1 and 2. 1, 1, 2, 2, 3, 3. You can see here 4, 4, 5, 5, 6, 6, like these guys. So you understand now what is an autosomal trisomy and what is a sexual trisomy. So Down syndrome, whenever you hear the term, it is called as autosomal trisomy. So what happens when you have an extra pair of a chromosome? See, guys, one thing that you have to understand, students, is there is a proper balance in the biology where you have the gene dosage. Okay, so the one chromosome and its homologous pair, the another chromosome. For example, you have 1, 1, 2, 2, 3, 3. Like this, you have up to 23, 23. No, for example, 22, 22, and then you have 23, sorry, 22, 22, and you have 23, 23. So this is called as a gene balance. There is a gene, and then it has a counter gene. Whether it is a similar gene or a dominant or a recessive, we don't know. The nature can depend on what type of genetics it might exhibit. But you can understand that there is a clear balance here. So whenever this gene balance gets higher or lower in cases of monosomy or trisomy, our body physiology completely changes. So because there is an overexpression, I told you, whenever you have a mutation condition, you have two types of situation. Either you have an overexpression of a gene or an underexpression of a gene, or gain of function or loss of function. So this can happen. So when we talk about a higher exhibition or higher expression of a particular disease, so look at your Down syndrome. Okay, hold on. Oh, sorry, you have your Down syndrome. Okay, yes, I'm sorry. I think some of the slides are missing. Just give me a second. Just give me a second. No, please. It's not working again. There's a small technical difficulty, guys. Just give me a second. Okay. Yes, I'm sorry. I'm sorry about it. A lot of classes going waste today. And hold on, I'll give you. Yeah, I hope you can see now. Yeah, sure. So thalassemia, everything is done. So we go for I Down syndrome. Okay. I think the Down syndrome is kind of, yeah, we have a Down syndrome. So I told you that the Down syndrome is an autosomal trisomy disease. So what is some of the symptoms or some of the physical features that you can observe through this expression of an extra gene is what happens is for the people who suffers from Down syndrome, they are not quite normal. They have furrowed tongue, they have short toes, short digits, okay, and they have a kind of carry bags or eye bags, and their face is very small, the neck is small, and these people have short ears, and they also have the disability, some of the mental disorders. Such as they don't have the they have a very poor cognitive abilities. Cognitive abilities is like thinking. For example, if I talk to you, you can understand me, right? So for them to understand and react, it is quite different from normal people. So they don't have, they're not mentally fit, okay, completely. So that is another thing. And other effects is the organogenesis, and there is a very poor organ development with these people who suffer from Down syndrome. So these are some of the side effects, I mean, like symptoms that they exhibit. So guys, I think you know the symptoms and it is in your textbook, so we're just going brief according to the syllabus, so you just follow what is there. Okay, next one we have is a Turner syndrome and the Klinefelter syndrome. So Down syndrome is an example for autosomal trisomy. Okay, and then you have other type, it is called a sexual monosomy, and then you have trisomy. So Turner syndrome is called as a sex chromosome trisomy or sex trisomy. Okay, so it is called as sex trisomy or sexual chromosome trisomy. So what is happening in Turner syndrome is, see, usually we have 44 autosomes and then we have sex chromosomes, either it is XX or it is XY. But in Turner syndrome, what do we have is 44 autosomes and then we have only one sex chromosomes with the absence of another sex chromosome, that is Y or X. So this is called as Turner syndrome. What happens in Turner syndrome is, so usually you can look at the person and then you can see more of feminine characteristics. Okay, you can see more of female characteristics. The appearance, everything would be more feminine, and the reason is X has a lot of genetic characteristics. X holds a lot of genes that codes for your body. That's why when you look at them, you look like more female kind of a thing, but their stature is very short. Okay, they are very short people. They're not as tall as you. Other thing. Second thing is they have a short neck and a webbed neck. Another thing, and they have a webbed hand, and they usually have poor organ developments with respect to their heart. Okay, and you can see here short stature, low hair, and then you have a wrinkled widely spaced nipples, and then you have shortened metacarpals. That is, the fingers are very short, and then we have short fingernails, and then you have brown spots near the navel, and then there are so many symptoms that comes with Turner syndrome. So whenever one particular sex chromosome is missing, so usually the whole development of the body is rudimentary. It's not properly developed. The organs are not properly developed. So that is called as a Turner syndrome. And next we have is a Turner syndrome. In Turner syndrome, we have sex chromosome trisomy, where we have XXY condition. Okay, and additional X chromosome joins during this particular situation. And usually both Turner syndrome and Klinefelter syndrome is due to the dysfunction. Okay, the major cause is the chromosomal spindle fibers. Okay, the spindle fibers do not actually properly separate these chromosomes during the gametogenesis. So that's when these particular situations cause. So what about in Klinefelter syndrome? It is called a super males or super females. The super females is another term we can usually use. So they look like male and they show the development of gynecomasty. Gynecomasty is the development of the breast in the male. Okay, this is called as gynecomasty. Next one is these both in Turner syndrome and Klinefelter syndrome, they are sterile beings. So the testes, the ovaries are not particularly developed. So they have a very tall stature. They have masculine voice. They have the masculine strength also. Their behavior and psychology is also like masculine. But some of them, they show a lot of feminine characteristics with respect to their their behavioral and social attitudes. Okay, and moreover, they show the development of mustache, they show the development of beard, okay, and the pubic hair and the rudimentary penis. So this is about Klinefelter syndrome, okay. So students, so we have finished the principles of inheritance and variation. So this was supposed to be a brief topic, and this particular topic is usually textual based. There won't be anything extra apart from the chromosomal numbers, whether it is monosomy or trisomy, okay, aneuploidy, haploidy, diploidy. So these conditions they will ask. So apart from that, nothing much you need to worry about these chromosomal or Mendelian disorders. And make sure you know how the genetics and how the pedigree chart works. Got it? No? So this is about the last chapter. So coming to today's topic. So today's topic is molecular basis of inheritance. Okay. So just 1 second. Mm, Still not present. Okay. Hold on. The only thing is I'm not able to annotate the ink properly. I'm really sorry about it. Okay, chalo. So when we look at molecular basis of inheritance, we understood that in Mendelian genetics, what happens is what are characteristics, what are alleles, what are traits, what is a dominant allele, what is a recessive allele. So if they follow some order like these dominance, recessive, and all those conditions, so we categorize them into Mendelian genetics. So sometimes what will happen, it's a reverse. You cannot even put it in a box. So you put it in the chromosomal disorders. So this was understood long before people understood mitosis and meiosis. So Mendel, he did a very great work. I told you he's a versatile genius. He understood something that many people did not even was not even able to give credit to it. They were not able to accept the mathematics in biology. So he was that far ahead in time. But now what happens is the human mind is very nice because we don't compromise just because we don't know the answer. We keep on searching for the answers. That is what we are. So now what happened is these people, the people who understood the Mendelian genetics, even though they agreed or not, they did not stop from looking for the genetic material. So what is the genetic material? Anything who carries the characteristics, who is responsible for an organism to have these characteristics. So just because Mendel mentioned factors, that doesn't mean that it will be a factor, correct, right? So it can be anything. So that is why people wanted to know what are factors, who is it, who is responsible, what is factor, what is the chemical nature of it. It is God made. So if you just go back a little bit and then if you see what did people believe in, then you will understand how much would be the fight to understand all the things that has happened through the history of time. Back in the days, they were like church. They believed that it is super, something like God. The God actually transferred the characteristics. So everything in the world was created as it is. Whether you slaughter many beings or you don't do anything, the creation exists as it is. So that was first belief. That means something magical is responsible for you to remain your character or whatever, whether you're human or a rat or a cheetah or a chimpanzee, whatever it is, that is cosmologically created. I mean, like cosmos are responsible. The God is responsible. So the one problem here is they said that the people who believed that special creation, that means theory of special creation. Here the people said that whatever is created here remains as it is, and how much ever million years it is, it's gonna be the same. So this is the Western thought. The Western people thought that it's gonna remain as it is. But if you see other civilizations, for example, Egyptian civilization and majorly our civilization in India, and if you see more of East civilizations, our belief is quite different. We believe in cycles of life. We believe in there was some life in the past. There were some beings in the past, which is not similar to today. So many of our scriptures, were Egyptian, Greek, and us, so we match in several points where we believe that there was life way ahead of what we see today. So this is what it is. You get my point? And some believe sperms are responsible for transfer of all the characteristics. Some they are called as spermists and ovists. Ova is believed to be a cradle of life, like a soap box, where she is responsible for holding the all the characteristics and transfer to the next generation. So this was another belief. And if you see another if you see another set of people, for example, the greatest believer in the scientific era during the 90s, early 19th century, was who you have your Charles Darwin. So Charles Darwin, what he did, in spite of him traveling to so many different continents in his own ship, okay, so he's a very beautiful example of being a perfect biologist, because he worked in fields. Many countries he visited, many islands he visited, and he saw through his own eyes what is life. Okay, how living organisms, what is how they evolved, how they become. So that's how the theory of evolution was suggested. People were believing Darwin more. So before Mendel, when Darwin suggested, you know what Darwin told? How are the who is responsible for the characteristics to come? He said that in male and female, okay, if you take male and female, these characteristics or whatever the nature you have, they're present as micromolecules. So these micromolecules, they come together from male and from female. They get together, they reorganize themselves, and it becomes a new baby. He told this, even though it makes sense. But was Darwin able to pinpoint to genes or factors or DNA? No, he was not able to do it. Then Mendel came and Mendel told that there is factors. So people started kept on searching. The people kept on searching what is an ideal genetic material or who is an ideal genetic material. Today, if I ask anybody, any one of you, what is there in your cell? Probably you might miss mitochondria or chloroplast or endoplasmic reticulum, but you will definitely come to nucleus. If I tell you what is present in nucleus, you will tell genetic material at least. If not, if you tell DNA or RNA, you will never tell DNA and RNA. Or maybe you will tell DNA and RNA. But many of you people will tell, sir, there are characteristics which is present in nucleus. Correct, right? That means you have an understanding. Do you think that this understanding was already pre-born? Definitely no. Today, if I ask you, what you will tell, what is the genetic material, guys? What is our genetic material? Some of you people will tell that, sir, genetic material is present in chromosomes. Some of you will tell that, sir, genetic material is present in nucleus. You get my point. Some of you will tell that, very smart guys, or I'm not telling smart and not smart. It depends on whether you heard the information or not. Some of you will tell that, sir, genetic material is DNA. Some people might confuse it to RNA or proteins. So let's look at what is that particular magic molecule, okay, which is responsible for giving us characteristics. Okay, see guys. So what is the genetics? For me, like what is this DNA? What is this RNA? If I look at DNA, if I take a bacteria and if I take any complex organisms that lie in between, whether it is a protozoan or a fungi or a sponge or any chordate or a non-chordate or a mammal or a non-mammal, anybody I take two examples and I put them together. What is the most common thing I can find? I can find one common thing is the genetic material, that is, sorry, excuse me. What is the most common thing? That is the DNA. Why my ink is not annotating, guys? I'm not able to annotate anything. Excuse me. Are you able to see the shift of the screen? Can you please confirm? No, not able to do anything. Excuse me. Yes, sir, visible. Yeah, my screen is visible, guys, but I'm trying to write something. I think you are not able to see that. That's my problem. Okay, hold on. I think now it might happen. Introduction class तो थोड़ा slow, then we'll fast up, okay? Yeah. So yeah, proper guys. I hope you can see the annotation. Just I put it line. If not, please put it in the chat box. Okay. So let's go for. I just told you. If I ask you guys what is the genetic material, finally, guys, you have to come to DNA. Okay, DNA is our genetic material. So what is DNA? So forget about the molecular composition. Forget about what it is, whether it is adenine, thymine, cytosine, guanine, etc., etc. You leave it. But if you see in a DNA, the whole number of molecules, different types of special molecules, the monomers are only A, T, C, G. Adenine, guanine, guanine, cytosine, thymine, okay, ATCG. So ATCG is what makes up your DNA. So yeah, what is ATCG, sir? These are nucleotides. I will tell you what is nucleotides. I will tell you how nucleotides become and how the construction of a DNA molecule happens. I'll tell you everything. But I just want you to imagine. Imagine a bacteria. Imagine a human being. Both of us have ATCG in our DNA. Only the rearrangement, only the arrangement of this ATCG in different ways, in different fashion, different numbers, etc., has resulted into so much variety in life that you see. Now, what do you call this? You call this a miracle. You call this the greatest possible uh, imagination that you can go to how to create things. Correct, right? If I have to ask you to write a computer code for your WhatsApp, for your Facebook, for your Instagram, how many thousands of codes you have to write, how many different types of letters you have to use, how many types of combinations of letters you have to use, how many commas, semicolons, and etc. you have to use to bring one particular app with minimum function. But look at the design of a living organism. Look at a tiger, look at a lion, look at a microorganism, look at anything that in your surrounding. How beautiful is their characteristics? How perfect are they? And how this perfection is maintained from one generation to another generation. If I have iPhone 11 or iPhone 13 or iPhone 15, does it evolve? You have a software update, guys, but you don't get an extra camera. You know what I'm saying? You don't get an extra camera. You don't get extra features. You don't get the faster processing. You know the science, but look at what's happening in the living organisms. Life started with a not it's non-cell. Life started with a no cell, and look how we are. In a normal human being, 30 trillion cells are coordinatingly functioning to make you what you are. So that is the beauty of genetics. That is the beauty of molecular biology. So we studying what is the reason for all these things, for all the we see, for all the characteristics that we observe, for all the diversity that we enjoy, and all the biodiversity that we envy. Okay, so today we will start this beautiful topic. This is my favorite chapter in the whole, not in the whole biology, but I mean to say this gives me an answer to why I chose biology. I hope you enjoy the same. So let's have an introduction, okay, to what is this molecular basis of inheritance. In molecular basis of inheritance, we are going to embark on a journey where we directly jump in with our already pre-existing knowledge that DNA is a genetic material. From there, we understand who proposed the structure of DNA, what is the search for genetic material, okay, and then we come to how the DNA functions, what is an RNA, what is a protein, and finally we understand what are we doing with this knowledge, like applications of molecular biology like DNA fingerprinting, criminology, etc., etc. It's a wonderful chapter. Let's get on to it. So, what's the time? We have 15 minutes. Let's go. Let's start here. Okay. So first the journey starts with nucleic acids. So we know that with the discovery of microscopy, people understood that there are microorganisms, and we are very curious beings. So as we examined more and more cells, we understood that either it is a plant cell or an animal cell, there is a centrally located isolated system. So the centrally located isolated system, finally, it was given, understood to be a nucleus. But usually, what is a nucleus made up of? It is made up of nucleic acids, which are macromolecules present in every living cells. So Friedrich Mischner was the first person to isolate these nucleic acids, and then he understood the chemical nature of the nucleic acids, and he gave a term called as nuclein. Okay, so when I said that he understood the chemical nature of the nucleic acid, that means he understood the pH. He got to know that it is acidic in nature. Okay, it is acidic in nature. Finally, once the discovery of acidic nature of nuclein was analyzed, finally all Altman called it as nucleic acids, but it was finally properly discovered and isolated by Frederick Michner. Frederick Michner was isolated and he understood the characteristics of it, and Altman only analyzed the acidic nature and he gave the term called as nucleic acid. Okay, so there are two types of nucleic acids that is found in every living cell or living organism. So that is either it can be a DNA or it can be an RNA. So many fantastic movies have been made in genetics. For example, X-Men, okay, Wolverine, or you can say Deadpool versus Wolverine. They're all mutants. So all the characteristics, all the Marvel, like for example Hulk. So if you see them, what do you see? You see they are not normal. So the abnormality comes with the change in the genetics. If you see old version, I'm an old being. So if you just see the old movies of Hulk, you can see that his father is a genetist and he tries to mutate his DNA and make him into a being who is indestructible. Okay, there it is. Okay, for science fiction and for everything, DNA, deoxyribonucleic acid. But this RNA is something, a silent killer. So the result of DNA, the expression of DNA is always carried through an RNA. We will understand what is an RNA and what is the significance of an RNA in the coming classes. So DNA you understand. So DNA stands for deoxyribonucleic acid. So what is a deoxyribonucleic acid? So if I talk about DNA, it is a genetic material which is found in all living organisms. Simple and plain. Sir, some of you will tell, sir, there are RNA viruses. What about them? Don't you say that? Why are you not saying that their genetic material is DNA or RNA? So why are you not saying that? If you ask me that question, you should know that viruses are not considered as a proper living organisms. They are a mimickers. Sometimes outside the host, they are non-living. Within the host, they are living. With this simple term, you can not involve or include the viruses under normal conditions. You understand my point? So what are living organisms from monera all the way till the complex eukaryotes? You can call them as all of us. Okay, all of us as a family. What do we have? We carry DNA as a genetic material. Are you following me? So what is a DNA? DNA, it is a code of information. I told you, DNA is a simple code. What is that code made up of? A structural arrangement, a molecular arrangement of four compounds, which we call it as nucleotides. They go by the term called as adenine, thymine, cytosine, and guanine. So this arrangement of four letters or four alphabets will give you the code of information. So this code of information is very much responsible for carrying any information. You know how much of information it is carried, guys. You need a supercomputer. Some 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 thousands of TBs of data is transferred. From one generation to another generation. It is simply fantastic. Why? Because you know that during the fertilization, it's only two cells that are involving in this whole process. Correct, right? So this coded information is traveling from one generation to another generation. Okay, not just traveling. We'll talk about what's happening between the process. So what is the structure? So it is a long polymer. When I use a polymer, so polymer is arrangement of monomers. A polymer is made up of when you have different monomers. A same type of monomers is called as homopolymer. A different type of polymer is called as, like for example, if I have glucose, C6H12O6 is what glucose. If I have many glucose C6H12O6 for n number of times, correct? No, for n number of times. Glucose, 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 glucose. Only glucose if I have rest. What do you call this? You call this a homopolymer. But what about DNA? DNA is also a polymer, but it is not a homopolymer. It is a heteropolymer. Why do I call it as a heteropolymer? Now, how I call glucose, glucose, glucose. It is not just made up of glucose, glucose. It is made up of nucleotides like that called as A, T, C, G. So since there is more than one type of monomer, we call it as a heteropolymer. Okay, so heteropolymer of what? It is a heteropolymer of deoxyribonucleotides. And four types of deoxyribonucleotides will come together to form a long chain of DNA molecule. So the volume of the DNA completely depends from one organism to another organism. So now don't tell me, sir, if we have more DNA, does it mean that we have extra characteristics? Does it mean that we are more supreme? Definitely no. It depends on how long the genetic chain has existed. For example, you can see some organisms where their ancestry is very, very long. For example, you can see crocodiles or monkeys, crocodiles, for example, crocodiles or amphibians. Their genetic volume is very, very high, even though they have less characteristics. Okay, human beings and elephants, if you compare the size, is very, very different. But does it mean that we have the gene number is like some millions in elephants and some thousands in human beings? No, the margin is very, very less. So the genetic volume can vary from one organism to another organism, and we cannot predict why this genetic volume varies. Okay, depends totally on the evolution. Okay, so this particular DNA, if you see the eukaryotic cell, okay, it is present in the nucleus region, even in the prokaryotic cell, but the nucleus is naked. You know that, right? So, which is a chief component of all the chromosomes, and it is also found in the gene that is the DNA, which is found outside the nucleus. You guys see, the outside the nucleus is called as extra chromosomal genetic material. So let me put it here, so that you don't miss what I'm telling. It is called as extra chromosomal, which is very important. So whenever I say extra chromosomal, that means they are the particular DNA which is not in the nucleus, okay, but outside. So if I have a DNA outside, so when will I give importance to something which is outside the community? When it has some role. That means the extra chromosomal genetic material which gives characters. For example, if you look at plants, plants are having what color, guys? They have green color. When the natural color you can see is green. Who is responsible for the greenness that you see in the plants? It is the DNA which is present outside the nucleus, which is in the chloroplast, etc. Okay. So the nature, that means the ability of the cell to synthesize energy, is what character? It is not in the DNA. It is not in the DNA which is present in the nucleus. It is in the DNA which is outside the nucleus, which is the mitochondria. You see that? So this DNA, which has a role, for example, antibiotic resistance nature in the bacteria, or the color of the leaves, or the energy metabolizing ability of the cells, all these characteristics are extra chromosomal characteristics. So this extra chromosomal characteristics comes from what? Extra chromosomal DNA. What is extra chromosomal DNA? Any DNA that is present outside the nucleus is called as extra chromosomal. Very, very, very important point that you have to understand. Okay. So, hold on, guys. Okay, we have time. So this is about extra chromosomal DNA. So can we understand what is DNA made up of? What is DNA? So DNA is like any other molecule. What is a molecule? You already know. For example, you can take sucrose, the sugar you eat. It is a result of a molecule. The oxygen you breathe, it is a molecule. So anything, a combination of elements is called as a molecule. So DNA is also a molecule. So DNA is made up of three different compounds. Okay. So here, what do we have? Each DNA is made up of three important things. One, two, and three. What is that? First one is a deoxyribose sugar. Second one is a phosphate group. Third one is a nitrogenous base. So these are the three key ingredients of the DNA. So if you don't understand, imagine that we are making coffee. If I ask you to make your coffee, what do you make? How do you make? You take milk, you take coffee or the brew of a coffee, which we call it as decoction or a coffee powder, like your Nescafe. And to that, what do you add? Sugar. So three components make one perfect coffee. Like that, for one DNA, we have three important components like deoxyribose sugar, phosphate group, and then we have nitrogenous base. Let's break down one by one. What are these deoxyribose sugar? What is phosphate group? What is nitrogenous base? Then we compile, we add, oh, deoxyribose sugar comes where? Phosphate groups comes where? And then where does the nitrogenous comes? So together, all these three, what does it become? And after becoming that, what does it lead to? So that is where we start from here. Now, I'm very skeptical, guys. Why? Because if I start now, I will end up with only deoxyribose sugar. Correct? No. So the continuity will get cut. So can we stop here? With your thing, I can stop here. Otherwise, I will finish at least deoxyribose sugar. You confirm. You give me the thing. Excuse me. Guys, I want you to put me in the chat box. Sir, continue. Then we can finish deoxyribose, taking another 5 minutes. Or we can go in the next class. We can go through completely in detail. We can start from the structure of DNA. What do you say? Make it fast. And as you do that, next class. Yeah, perfect. Yeah, I agree. We will have a proper continuity. We have a good introduction. So we'll go with a proper continuity. I have also reduced the whole textual knowledge. I'll make sure that I compile the information and give you more of need. Okay. Chalo. So this is a very, very important chapter. Please attend every single session, guys. Okay. Only few classes will be like this, and then we'll switch to the studio. There we'll have a proper everything like the last classes. Okay. Chalo. And before you leave, please give me the feedback. Okay. I will. I'll just launch the poll. Just give me a second. Yeah, the poll is live. Please give me a feedback, students. It'll be very helpful, and I'll see you tomorrow. Thank you very much.